Water and photosynthesis. That is the role of water in photosynthesis. In class 7, we already studied how von Helmont found that water is essential for the increase of plant mass. Plant mass is increased. That is, mass means the trunk, the fatness of trunk. That is called mass. The leaves, they add to mass, and even the roots also are part of mass. So water makes water essential for the increase of plant mass. If there is no water in the plant will shrink to its some 90% size, 95%, because everything in any living substance is 90 to 95% is water only. So it makes the mass. He did not know about photosynthesis then. That means this von Helmont was not knowing anything about the photosynthesis. Later it was found that increase in plant body mass or material occurs due to the process of photosynthesis. We shall study more about it in the following sections. Read the chapter on nutrition in plants in class 7. Discuss with your friends and write a note on von Helmont's experiment focusing on how he concluded that water was important for plant growth and increase in body mass. That is about water. It increased the mass of a plant or any living organism. Air and photosynthesis. Just now we discussed about water and photosynthesis. Now air and what is the role of air in photosynthesis. Let us discuss an experiment on photosynthesis. We will discuss the experiment on photosynthesis. We have studied some experiments in our earlier classes about photosynthesis. This one helps us to find out about the role of air in the process of photosynthesis. It is interesting to learn about the experiment, which was one of the several milestones in the gradual development of our understanding of photosynthesis. It is interesting to learn about the experiment, which was one of the several milestones in the gradual development of our understanding. That we understood photosynthesis in our gradual advancement of age, not at once, but as the time passed and as the experiments on photosynthesis, especially on air, was conducted. Joseph Priestley, 1733 to 1804, in 1770 performed a series of experiments. In the age 1770, he performed series of experiments that revealed the essential role of air on the growth of green plants. And it revealed the experiments that were conducted revealed It means showed, revealed means showed the essential role of, that is the most important role of air on the growth of green plants. That air is essential in the growth of green plants. Photosynthesis was still not known to scientists at that time. At that time, people were not knowing all the process of photosynthesis. You may recall, oxygen was discovered by Priestley in 1774. Priestley was a scientist. In 1774, he discovered oxygen. The name oxygen was coined later by Lavoisier in the year 1775. When Priestley discovered oxygen, he did not name that gas as oxygen. It was another scientist. His name was Lavoisier. In the year 1775, he named that gas as oxygen. Priestley observed that a candle burning in a closed bell jar, this is bell jar, glass bell jar, a candle is glowing in it, is not? Priestley observed that a candle burning in a closed bell jar soon gets extinguished. If you light a lighting candle, burning candle, if you keep in a bell jar and close the bell jar, after some time the flame of the candle will be blinking and diminishing. Why? Because oxygen in the bell jar is being consumed by the burning candle. Soon gets extinguished. That means that there will be no more burning, that will get extinguished. Similarly, a mouse would soon suffocate in a closed space of the bell jar. If you keep a mouse, rat, in the bell jar, it will be respiring in the bell jar, taking oxygen and leaving carbon dioxide. After some time, all the oxygen in the bell jar will be finished, taken by rat or mouse. Then the rat or mouse die because it needs oxygen in respiration to live, to survive. Similarly, a mouse would soon suffocate. Suffocate means the respiration is slowing down, forcibly slowing down, is called suffocation. Similarly, a mouse would soon suffocate in a closed space of the bell jar. He concluded that a burning candle or an animal both somehow damage air. That he came to know that a mouse or a burning candle damages the air by consuming oxygen in it, and the air becomes less weight, the ratio of the gases in the bell jar will be lessened because oxygen will be over, either consumed by the mouse, that is rat, or candle, burning candle. Understand? Both damage air. When he placed a mint plant in the same bell jar, mint plant, mint means pudina. Mint plant he kept in the bell jar. He concluded that a burning candle, when he placed a mint plant in the same bell jars, he found that mouse stayed alive and the candle, when lighted from outside continued burning in the presence of the mint plant. He found that if you allow, there is candle inside the bell jar, and the oxygen in the bell jar is consumed by the burning candle, but that burning, the oxygen will be finished because of the burning flame of the candle. But if we keep a mint plant, a green plant, inside the bell jar, again the candle will be glowing, burning. Why? How? The plant takes carbon dioxide and emits and excretes oxygen from its economy, from its leaves and trunks. And that oxygen is essential for a burning candle. You understand this? Mint plant. Priestley hypothesized as follows: plants restore the air. What breathing animals and burning candles remove. Then he said that plants restore the air. That means there is a defective air. That is, oxygen is over by a burning candle. Air sans oxygen, air without oxygen. So that is what he called plant restore the air. Again the mint plant emits or excretes oxygen, what breathing animals and burning candles remove. He said that the burning candle are the breathing animals, that is, a rat is consuming oxygen and taking away all the oxygen. So he said, plants restore the air. That is, again plants bring the oxygen again in the bell jar with the help of their respiration. They take carbon dioxide and leave oxygen. And that means they restore. There was a defect in the air that burning candle or the rat had taken away oxygen. That was defective air, not good air, you can say, simple language. Now, the mint plant is kept inside the bell jar. Mint plant emits, it excretes the oxygen, and the defective air becomes effective air. What had Priestley done to introduce a mint plant without disturbing the experimental setup? How did he light the candle from outside? How he lighted the candle from outside? We have to know that. Do you find any relationship between candle, rat, mint plant? Discuss. Is there any relationship between? Do you find relationship between candle, rat, and mint plant? Any relationship is there? Priestley's experiment confirmed. Confirmed means एक पक्की बात बता दी, पक्की बात बता दी. Confirmed, consolidated. Priestley's experiment confirmed that gaseous exchange was going on and plants were giving out a gas that supported burning and was essential for the survival of the animals. What he observed is, and Priestley's experiments confirm that the gaseous exchange, the gas in the air was taken away. That gas was oxygen. Where it was taken away? It was consumed by burning candle and rat or mouse. And again that came back when you keep mint plant also, that leaves out oxygen and it makes you complete air. But how do plants take in air and utilize carbon dioxide for photosynthesis and oxygen for respiration? But how do plants take in air? How the plants take inside their body air and utilize carbon dioxide? We leave carbon dioxide and we take oxygen. The carbon dioxide left by the man is present in the atmosphere, and that will go to the rat. That will go to the plant. How do plants take in air and utilize carbon? The plants take the air and they utilize the carbon dioxide inside the air for photosynthesis and oxygen. For photosynthesis and oxygen for respiration. Plants take carbon dioxide from the atmosphere to bring photosynthesis in the plant leaf. And for the oxygen also is there, that oxygen is for respiration. How do they make the choice? Massive amounts of gaseous exchange occur through the stomata. How do they make the choice? Massive amounts of gaseous exchange, that we are giving out carbon dioxide and taking in oxygen, and this what is called productive gases. Massive amounts of gaseous exchange occur through the stomata. There are on the plant there are small pores. Pores means surah. We cannot see. We can see with a this electronic microscope. We can see easily. So we find in a leaf small cells. Massive amounts of gaseous exchange occur through the stomata. There are stomata in the plant. Stomata means it is bulging, usually present in leaves as long as they are open. When the leaf is open, the stomata, that is small holes, they are also open. While plants also carry on gaseous exchange through loose tissue and respiration. How do they make the choice? Massive amounts of gaseous exchange occur through the stomata. It is gaseous exchange, carbon dioxide, oxygen, all these going inside the leaf and coming out of the leaf. This is called massive amounts of gaseous exchange occurring through the stomata, usually present in leaves. Stomata means small holes in the leaf. Through this, they take respiration. Usually present in leaves as long as they are open. While plants also carry on gaseous exchange through loose tissues and stems. Not only in this leaf, but also in stem. Stem means from where the plant originates. That is a wood, some wood. Next to the stem, branches, then leaves, then flowers. How do they make the choice? Massive amounts of gaseous exchange occur through the stomata as long as they are open. While plants also carry on gaseous exchange through loose tissues and stems. While plants also carry on Plants also carry on, leaves also carry on, we have already told how they lived. In plant there are trunk also, you find trunk, that also stalk. For small plants we refer to as small, not massive. It is actually the level of the organelles involved in the process of photosynthesis and respiration that the choice of the gas is required. It is actually at the level of organelles involved in the process of photosynthesis and respiration that the choice of the gas required is made. That means the seeds and it is actually at the level of organelles. Carbon dioxide is necessary for photosynthesis. We have talked about air. Now we are talking about carbon dioxide. It is also necessary for photosynthesis. We need a de-starch plant to start with. We need a plant which is de-starched. That means the natural starch present in the plant is brought outside. That is de-starching. That is called while plants also carry loose tissues, we need a de-starched plant to start with. We will take a plant which has no starch, de-starched. That means all the starch is removed from the plant. De-starched plant to start with to make the experiments of photosynthesis we need in Mahal. This is very important. It is actually at the level of the carbon dioxide is necessary for photosynthesis. We talked about air and water. Or now carbon dioxide is necessary for photosynthesis. We need a de-starch plant to start with. Now to understand how the carbon dioxide is necessary in photosynthesis, we have a plant de-starch that contains no starch. For de-starching, we need to keep the plant in the dark for nearly a week. If we keep a plant in dark, for nearly 1 week, you have to keep in dark a plant 1 week in a corner. That even in daytime that must be dark. What will happen when the photo is not coming, that is sunlight is not coming? Photosynthesis program is stopped. You understand? We need a de-starch plant to start with. For de-starching, we need to keep the plant in the dark for nearly a week to remove the starch. That is, the starch is removed from the plant when we keep the plant in the dark. If we focus light on the plant, we know there is what is called, we need a de-starch plant For discharging, we need to keep the plant in the dark for nearly a week to remove the starch from the leaves. Arrange the apparatus as shown in the figure. Take a wide-mouthed transparent bottle. Take a wide-mouthed transparent bottle. D-starch means divide of starch. That is, that plant contains no starch. Starch means carbohydrate. Put potassium hydroxide pellets or potassium hydroxide solution in the bottle. Potassium hydroxide is a chemical. It is found when you pour some potassium in water, in a half glass of water, potassium hydroxide is formed. A new compound, neither potassium nor water. Potassium hydroxide is an entirely different compound formed by chemical reaction. The chemical reaction came because the potassium was dropped in the water. And potassium hydroxide is made like that. Put potassium hydroxide pellets. Pellets means, what is the meaning of pellets? Pellets means goliyan. Small tablets. We take a crocin tablet, You make 4 tablets, 4 pieces in one tablet, crocin tablet. Each small tablet is called pellet. You make, first of all, potassium hydroxide and make pellets, small pieces, with the help of water. Potassium hydroxide absorbs carbon dioxide. Potassium hydroxide in general दुकान जरूरत नहीं है, कहाँ तो खुली जगह में रख डाले तो अच्छा बाहर चले आते थे उसके पास. Insert a splitted cork in the mouth of the bottle. We have kept the plant inside the bottle and after a few hours, after some time, after we have tested this leaf and any other leaf of this plant for starch, as mentioned in activity. Insert splitted cork in the mouth of the bottle. Splitted cork. Splitted means 2 pieces, made into 2. Splitted, तड़क को रहता, कारक रहता. थोड़े बॉटलों को जो ढक्कन नहीं लगाते, कारक लगाते. Splitted बोले तो 2 टुकड़े, 3 टुकड़े, वो कारक है उससे. Put potassium hydroxide pellets or potassium unnamed plant in this plant. The cork in the mouth of, insert one of the leaf of de-starch plant. Through a split cork into transparent, that means there is one plant and there is one small bottle. The bottle is covered, closed with a cork. That cork is two-piece cork. Splitted means two-piece cork. And in the split, that is in between the two pieces, we keep the leaf. This leaf and any other leaf of this plant for starch, as mentioned in activity 1. Then we will see the starch. Where is the starch? We can see. The quantity, the area of leaf we kept inside the bottle will not get sunlight. That will be in the water. So the leaf part outside the bottle will be having starch. You know how we keep the leaf inside the splitted cork? You know that? Suppose this is cork. This cork closes the bottle. Bottle close. So with the help of a knife, we make a split. Split means cut. Cut this cork into two. And those two pieces insert. रख डालो, स्टार्च इंसर्ट कर डालो बॉटल में, दो पीस भी रखते टाइम को ये पत्ता भी रख डालो. अब धूप में रखो. धूप में रखे तो क्या होता, अंदर के पत्ते को वो इसके बीच में रहता ना, इसके कार्क के बीच में, उसे धूप नहीं लगती. धूप नहीं लगी तो उतने दूर दैट मच, जितने दूर जो है हमें कार्क जो है लीफ को कार्क के अंदर रख डाले, फोटोसिंथेसिस होता नहीं. बाकी के पत्ते में फोटोसिंथेसिस होता. बाकी के पत्ते जो है स्टार्च आ जाता उसमें, और ये हमें जो बंद कर डाले उसमें जो है स्टार्च नहीं रहता, डी स्टार्च. स्प्लिटेड कार्क बोले तो कट कार्क, कटिंग. स्प्लिटिंग है तो कटिंग, दो पार्ट में कट कर डाले कार्क को. कार्क you have seen कार्क. कार्क is used for closing the bottle. पुश करके क्लोज करा लेते, ऐसे थ्रेड्स नहीं चढ़ाते. पुशिंग. मेरे नज़र ऊपर अपस्टेयर वो अलसी का तेल रहता है बॉटल में, उससे कार्क रहता नहीं, यू नो सीन. कंप्यूटर के ऊपर है, अलसी का तेल डाल कर छोटा ऐसे चौड़ा बॉटल, ऊपर उससे कार्क लग कर रहता, कार्क बोलते. उसके दो टुकड़े कर डालते, दो टुकड़े करके बीच में लीफ को रखते. अब जो है वो धूप में रखे तो जितना पत्ता जो है कार्क के अंदर है वो फोटोसिंथेसिस नहीं होता. बाकी के जो है वो फोटोसिंथेसिस, बाकी का टुकड़ा लीफ का है उसमें फोटोसिंथेसिस हो जाता. Insert one of the leaf of discharge plant, transparent bottle containing potassium hydroxide. बॉटल में क्या रखो है, पोटेशियम हाइड्रोक्साइड रखो है. पेलेट्स पोटेशियम हाइड्रोक्साइड सॉल्यूशन. लीव द प्लांट इन सनलाइट. सनलाइट में रख डालो प्लांट को. कार्क में रखो पत्ता सब सनलाइट रख डालो. आफ्टर ए फ्यू आवर्स, 2-3 घंटे या 4 घंटे के बाद क्या होता, यहाँ फोटोसिंथेसिस आता पत्ते में. जिस जगह कारक से बंद करे नहीं, धूप जाती है सब जगह, वहाँ सब फोटोसिंथेसिस जो हरा भरा है पत्ता. जिस जगह जो है वो धूप नहीं जाती है, कारक बंद कर डालकर, वहाँ हरा भरा नहीं रहता, स्टार्च नहीं रहता ना. यू अंडरस्टैंड? Leave the plant in sunlight. After a few hours, test this leaf and any other leaf of this plant starch as mentioned in activity 1. The leaf part which was exposed means exposed, this is exposing, what is this called? This board is called the sunlight. The part which was exposed to the atmospheric air becomes bluish black. The leaf part which was exposed to the atmospheric air becomes bluish black, and the one inside the bottle containing potassium hydroxide which absorbs carbon dioxide in the bottle remains colorless. So that carbon dioxide necessary for photosynthesis. The carbon dioxide works on the plant leaf. That's why the leaf is green, but the leaf is not green, the part of leaf which is inside the cork. We cut the cork and keep the leaf inside the, in between the two parts of the cork. Why was the plant kept in dark and then in sunlight? You tell me. Because when we keep the plant in sunlight, photosynthesis takes place, starch is formed in the leaf. Starch means carbohydrate. Glucose also called starch. What was the plant? Why was plant kept in dark and then in sunlight? Afterwards, we kept that leaf in the sunlight. When we keep the plant in sunlight, the sun rays come and take care of the leaf. Understand? That is, sunlight is necessary for photosynthesis. The leaf part, which was exposed to the atmospheric air, becomes black, and the one inside the bottle containing potassium hydroxide, which absorbs carbon dioxide in the bottle, remains colorless. There is no color. The part of the leaf which is inside the bottle, it has no color. Colorless it becomes. But the leaf part which is outside the cork becomes blue-black. We have so far discussed the role of water and gases in the process photosynthesis. Scientists who had been working on these lines had observed some other factors that affect the process of photosynthesis.